Yep. So, a direct welcome to all of you present here. This is Shri to this World Pharmacy Day. Good morning. Myself Shubhi, host of the day, and it's my absolute pleasure to welcome you all to today's event. I would like to call upon stage respected Professor Murthy Sir to invite Honorable Dr. Konark Nicholas, scientist from University Life Indiana, USA. Econ sir, we are audible. Econ sir, we are audible to you. Econ sir, we are audible to you. Hello, Dr. Nicole sir, we are audible. I'm trying to get my screen set up here. So good morning, it's the first time I've used Google Meet. I'm trying to get my screen. My apologies for issues. First time I've used Google Meet.
Nah, itu catat ya. Tentu tak akan dari perlu bring doctor this one the was bring down firstly numbers either scientists today and because that university will be together going to give the presentation mother science work so on behalf of the university welcome you. Thank you. Yes, kawan colleagues आणि संख्या मैंने डायरेक्ट हमसे he saying comfort ये era कहाँ से अरे पूरा यूनिवर्सिटी era
Okay, my apologies. I sincerely apologize for the technical difficulties. My apologies. I sincerely apologize for this selection. All right. Technical difficulties. Okay, you everyone see my before page. The front, the first slide, everyone see that? Okay, the front and the first slide.
Community feedback, yes or no?
Yes, sir, screen is visible too.
All right, sounds good. Okay, started then. My apologies for the these are because I'm all having a great morning. Maybe it's my plague viruses, particularly skin disease. Now, these two viruses are quite endemic in most of the world. I won't get into all that here in a moment, but there's no drugs available to treat these viruses. So the main issue for me was better understanding the structural and biochemical assembly mechanisms of these viruses to then identify locations to develop drugs against. So, as the title of my talk today suggests, we'll be talking about plague viruses. These are, in fact, a global phenomenon. Now, these viruses are transmitted by mosquitoes, particularly of the Aedes genera. There are several as well transmitted by ticks and unknown vectors we've been able to find in examples of flaviviruses in the oceans, in starfish, in mollusks. So they're quite well diversified group of viruses. Now, in humans, the symptoms of these viruses range anywhere from fever and rash to meningitis, even encephalitis, hemorrhagic fever in the case of dengue, and microcephaly in the case of Zika virus. Altogether, there's about 4 billion people who are at risk of flavivirus infection each year. And so there's a significant need to develop antiviral therapeutics capable of targeting ubiquitous mechanisms across most, if not all, flaviviruses. Now, the reason I say most, if not all, flaviviruses is because of their unique structure. Flaviviruses share a high degree of structural homology across all of the different species. And as you can see here, we have the Zika virus over here on the left, looking down its fivefold axis, dengue virus serotype 2, and West Nile virus here. And all of these show that high degree of structural homology. Not only do they share structural homology, but they are many different single particle reconstructions, both from the lab and elsewhere, show this a high degree of homology between different species, but this suggests a much deeper similarity between each of the viruses in their genomes. So, of course, the flavivirus genome is a positive single-stranded RNA genome that encodes two different groups of proteins. The first is the non-structural group of proteins. These seven proteins, NS1 through NS5, function primarily in the replication, assembly, and immunoevasion aspects of the virus life cycle. While the structural proteins containing capsid protein, pre-membrane or membrane protein, and envelope protein are all there to form the actual virus structure. And these are the proteins that we'll actually be taking a look at today with specific attention paid towards their transmembrane heats shown down here. So we know quite a bit about the flavivirus life cycle. We know that the flaviviruses can attach to cell membranes and they're brought in different mechanisms. We know quite a bit about their uncoating process, how the actual envelope proteins fuse the membrane of the endosome to the membrane of the virus itself. And then we know quite a bit about the virus maturation and the pathway of how the virus goes from this spiky, immature structure to a smooth, fully mature virus that is ready to go on back to new cells. However, very little is known about the flavivirus assembly process, how actually the nucleocapsid core, shown here in purple, how that core mixed with capsid proteins and RNA actually identifies the glycoprotein-rich regions on the endoplasmic reticulum membrane, and how does it form particles. We haven't been able to show definitive evidence yet that the core of these particles are are an actual structure. They seem to be just kind of this ball of spaghetti or noodles with different kind of proteins scattered throughout them that somehow buds into a virus particle. But how does that, what interactions are there, and can we actually identify those interactions to then treat them like a druggable target? So before we get too much deeper into the brain virus assembly pathway, something that is quite interesting with these viruses that we haven't been able to fully understand as of yet is why they remodel a huge portion of the cells that they infect and the membranes within those cells. So you can see here up at the top in these electron micrographs, this is a cell mitochondria shown here in the end. You can see the cristae, the folds of the membranes of mitochondria quite. But when you infect the same kind of cells with dengue virus or dengue virus serotype 2, you get these massive membrane structures, these structures called convoluted membranes, that can send out these little tendrils that touch mitochondria, that touch the endoplasmic reticulum, and even the nuclear membrane. And there's this massive exchange of these different lipids that are occurring between these convoluted membranes and the mitochondria and other membrane surfaces. And indeed, when we look at electron tomograms, what we can see here is in the uninfected cells, we have nice puncta or nice little pockets of membranes showcasing the mitochondria here. But then when we actually infected viruses, we infect the cells, we get what is called the spaghettification or the elongation of these mitochondrial membranes because there's some need for massive amounts of lipids to be transferred from one area of the cell to another to encourage virus production. And so there's this interesting interplay between lipids, the virus proteins, the host proteins, and the construction of new virus particles to then bud off out of the cell that is occurring all the way through virus infection. And so if we can identify how all these parties play together, we can probably find some unique drug targets. And so before we get into all that, though, kind of just a refresher on what are the building blocks of membranes, in case there's some of you who are unfamiliar in the audience, as well as others who would like a little refresher. So the three main parts of a membrane are phospholipids. These include phosphatidylcholine, ethanolamine, serine, glycerol, and inositol. These are just a phospholipid head group with a lipid tail. And we can see down here broken up into the different percentages of what between mammals and insects, how these different membranes differ. And something interesting about flaviviruses is because they infect both mammals and insect cells, and there are certain lipids that are unavailable in both species, but these viruses proliferate quite well. So they've been able to kind of master the art of building membranes out of different starting materials to be just as varied. And so another major aspect is cholesterol. And cholesterol, of course, functions to kind of squeeze membrane lipids together, to pack them tighter together, and to create raft structures on the membrane to allow for different kind of protein-protein interactions or protein-lipid interactions. The last group is ceramides. And ceramides are similar to phospholipids in their structure. They have two long tail groups, but they lack that large phosphate head on the top of them. So these are the three main groups. But what happens again with flavivirus infection is there's a lot of reconstruction. And so we can see here that these different lipids have different properties, allowing them to function differently within the membranes. So we have our phosphatidylcholines, for example, shown here. I'm actually going to change my pointer. Phosphatidylcholine that as actually can't give any curvature to the membranes. These make really nice flat lamellar phase membranes while the inverted or conical lipids, such as phosphatidylethanolamin, can actually induce what's called negative curvature. And so when we're talking about a virus budding off of the membrane, that membrane has to go from a flat phase to something that is highly bent. And that bending process requires the improvement of specific lipids to allow for that bending. And so these phosphatidylethanolamines, these inverted conical lipids actually induce negative curvature, while phosphatidylinositol actually induces positive curvature because it's a conical. We have one that is inverted up and one that's inverted down, and these two function together to form both leaflets of the membrane to help with the virus flooding. Cholesterol now can actually compact membranes. And so we can see here is in a liquid a liquid phase. What we can see is that all of these side chains are kind of spread out with the different lipids. When cholesterol is introduced, it can kind of compact those lipids tighter together, so you can fit more into a tighter phase. When you then all of a sudden remove those, you actually get a even tighter gel phase forming. And so cholesterol can actually help to make membranes more fluid or more rigid, depending on how they're being used by the surrounding proteins. And lastly, the ceramides can kind of do a little bit of both. They can kind of sit into membranes. They can induce what's called membrane flip-flopping. So they can encourage certain lipids to transition into one leaflet or another, and they can actually induce negative curvature. So curvature away from the cytosol is what a negative curvature is. And they can also form domains, so like little islands in an ocean of very fluid lipids. Those islands are a bit more stable. They can allow proteins to hang on to those specific lipid groups. Now, this is all a lot of information about how lipids work, but this is important because the lipids need to do their own little jobs to allow for virus budding. And that's something that I was highly interested in looking at is how the interplay between these two groups, proteins from the virus and lipids from the cell, play with one another. And so we can see here in this electron tomogram is what's called the building of replication complexes during plague virus infection. All of these spherical structures that you see here in brown or tan, all of those are replication complexes of non-structural proteins that are actually building new pieces of the virus RNA. Now, that viral RNA is packaged into these little red holes, which are the actual virus particles. What we've been able to see is that the construction of these replication complexes occurs very closely to those virus particles. And they happen almost one to one. And so with one replication complex, a virus buds off nearly right next to it. But what this requires is a lot of membrane compacting. To allow for membrane bending to such a degree, we need lots of cholesterol. But something that is well known within the field is that cholesterol is mostly found within the plasma membrane. However, when we actually look at the transfer of cholesterol from the outer membrane, so shown down here in these mock samples, you can see how all the cholesterol that has been tagged with these fluorophores is actually found on the plasma membrane. When you infect with West Nile virus or dengue virus also, all that cholesterol is taken away from the membrane, the plasma membrane, and transferred into the pockets of these replication complexes and viruses as well. And so there's this biasing effect, and cholesterol is being moved from these plasma membrane to sites of replication. And because replication complexes and virus assembly are happening in very close proximity to one another, it was hypothesized that cholesterol is a highly functional and much needed component of virus assembly. Even more so when we kind of look at all of the different lipids being made within the cell during flavivirus infection, we can see that all of them are upregulated, meaning that the viruses upon infection actually turn on the cells to make more lipids of a specific species. And so phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, all the ceramides, sphingomyelin, all those pieces are being regulated to make more of them. However, cholesterol kind of stays the same, and that just is being trafficked from the from the plasma membrane to the surfaces of the replication complexes and virus particles. Now, this is all interesting, but how does it relate to the actual virus membrane itself? It's one thing for the virus to induce all of these different membrane structural rearrangements to encourage cholesterol to come from the plasma membrane to sites of replication complexes. But what does it mean for the virus itself? What is the membrane of the virus particle actually made of? And a lot of studies have looked at this using single particle analysis, lipidomics. And what they've been able to find is that ceramides are highly upregulated within the virus membrane. Excuse me, they're highly enriched. So much more ceramide is found within the virus membrane compared to the cell membrane, as well as the negative lipids, phosphatidylserine, glycerol, and inositol are up regulated or highly enriched within the virus membranes compared to cellular membranes. And phosphatidylcholine, which is the primary building block of pretty much all membranes within cells, that is the most abundant form of lipid, is actually downregulated within the virus membrane. So what this suggests is that there's a biasing effect towards negative lipids, lipids that contain a negative charge within their phosphate head group, and ceramides, which actually help to bend the membrane, and cholesterol. So there's this kind of picture being painted that we're biasing towards these negative lipids. We're not really sure why. So altogether, to kind of summarize a bit, we have a decrease in phosphatidylcholine. We have an increase in all of these negative lipids. We have a significant increase in cholesterol and a significant increase in ceramides. So what this suggests is that the proteins within the virus themselves, within the virus particle, so the structural proteins, PRM, E, and capsid protein, might be playing a role in manufacturing the correct lipid environment necessary to then produce virus particles. And this was something fascinating for me because wanting to go into drug development, and I know there's a lot of drug development people in the audience today, and maybe even some pharmacists as well. This might seem very, very fundamental, very basic research. Okay, like it's great that we know how the virus puts itself together, but how can we make a drug to combat it? It's a great question to ask because if, for example, the virus highly depends on cholesterol to be transported from the membrane to the sites of replication, there's been studies showing that cholesterol drugs or drugs that actually decrease cholesterol within the plasma membrane can actually combat and actually lower the infection duration of flaviviruses. And so just understanding pretty much the basic fundamentals of how these viruses assemble can teach us new pathways, new drug pathways that we might not have experienced or explored before. So that was an eye-opening experience. But to take all this into together, I want to kind of discuss a little bit about the assembly mechanisms of these viruses and how they put themselves together using their virus protein. And then we'll kind of bring in the lipid equation together at the end. So right here, what we have is on the left here, a replication complex where we have all of those non-structural proteins that I mentioned before. They're producing new viral RNA from the copies of the double-strand RNA that they have. And there's this protein called NS2A that can actually latch on to the RNA and transport it to sites of assembly. Now, these NS2A proteins also function as a chaperone to bring in new pieces, all of these unprocessed polyproteins that we talked about earlier, and they can actually bring them to those same sites. And so NS2A almost actually acts like a chaperone, bringing in RNA on one side and proteins on the other to all of the same location. And of course, we don't really know how the bending starts. We don't really know how capsid is able to identify the specific region to bind to. I will get into that soon. But overall, we're able to kind of piece these ideas together that all of the components that are necessary for virus assembly have all been brought to the same area. And this was a key insight that was identified back in 2019. Also, assembly mechanism number two is that capsid itself can actually have an organizing ability of the glycoproteins. And so what I mean by that is capsid protein is usually in the monomer. That monomer, though, can actually dimerize on itself. And that dimer actually has been shown to bind very tightly to RNA. And so the existing paradigm suggests that capsid is able to form out of monomers, it forms dimers, those dimers then bind to RNA. However, in a recent publication back in 2020, a group was able to find that some population of capsid is able to retain what is called the alpha 5 helix. It's not important to remember overall, but there's an extra piece holding on to these capsid dimers. That extra piece is actually able to form these oligomers, these trimeric oligomers, that actually function to organize the glycoproteins on the membrane surface. Because there's kind of two components happening at once. There is the formation of the nucleocapsid core, this ball of capsid proteins mixed with RNA. But then there's also all of the glycoproteins spread out along the endoplasmic reticulum membrane that have to be brought together. because flaviviruses have a very high degree of symmetry within their structure, and that symmetry requires a high density of glycoproteins on the virus surface. In fact, there's 180 copies of the E protein in almost every single virus. And so there has to be some kind of organization ability taking place throughout the assembly process. And what this group found was that capsid, using these two different populations of itself, was able to function as an organizing ability. However, we don't really see any interactions between capsid and the transmembrane helices of PRME. And so there seems to be this missing link. How does capsid, in fact, find these proteins, and how does it organize them? This was a nice step forward in understanding flavivirus assembly, but it didn't tell us the full picture. And so that's where some other data has shown that lipids might play a role. And in fact, capsid was able to bind to highly negative regions within a membrane patch. And so what they did was they took capsid proteins and they washed negative lipids. Those capsid molecules were actually to stick to them. And because capsid itself is highly positively charged, I should have prefaced that, it's a very positive protein and it sticks to these negative membrane patches. And when you wash over the RNA is actually able to bind to the other side of the capsid, pretty much facilitating a kind of chicken and egg problem. Its capsid can bind to either the RNA and then to negative lipids or to negative lipids. Overall, it gives a nice kind of understanding. It's like, okay, we have this ball of capsid and RNA, and that ball of capsid and RNA can find its way to negative regions within the membrane. But again, we're missing this crucial link here. It's a small step of one, how does that negative membrane region form? What is encouraging the formation of that? And how can the glycoproteins and capsid themselves all interact with this negative region? And can we identify how that region forms? And so that's where my work took place. Is again, because all the components are within that same location. The lipids and structural proteins and the RNA are all located together. But how, of course, do those two pieces stick together? Can we get an idea of how capsid is attracted to the membrane, these negative regions? And can we understand how those negative regions form? And that's what I wanted to look at. And so some previous structural studies from my lab have shown that both the mature structure of Zika and the immature structure of Zika show a lot of similarities. We have our envelope protein transmembrane helices, the lipid bilayer, and even the nucleocapsid core shown in magenta. However, in the immature structure published in 2017, we were able to see for the first time that there was this leftover density. Some density here that wasn't showing up in the mature structure and hadn't shown up in any of the previous immature structures either. And so what was this residual density? So my job was to kind of look into this phenomenon. And what we can see here is a cross-section of a Zika virus immature structure. And what we can see is this residual density fits capsid protein not very well. The structure is not well defined. It's very low resolution. But overall, this globular structure was able to fit most of the atoms of the capsid protein within the electron density map. But what were the structures above? Of course, the transmembrane helices, but from where? Which proteins were they from? And so when fitting in the trimeric spike or the immature trimeric spike, we could see that those transmembrane helices didn't really work. However, when we look at the adjacent proteins and we kind of get rid of the noise and we focus only in on those transmembrane helices, we can see that those fit quite nicely within the structure itself. And taking a closer look, what we can see is that within the PRM protein, so right here is just the transmembrane helix, the anchor of the whole protein. I'm actually going to go back here. What we can see is that in these massive structures, you have E protein up here, PRM is shown here in blue, and the E transmembrane helices is shown in red. So we have two groups. We have a PRM protein and a E protein. Both of those can function together in what's known as a a heterodimer. So for every E protein, we have one PRM, and they stick together. So they kind of two proteins that stick together and function as a single, pretty much. But what we can see is that there's three copies of each of them directly above the capsid protein. And those three copies of both E and PRM were in very close proximity to the capsid protein, hypothetical capsid protein density. And so again, taking a look at what is around those regions, we can see that the PRM transmembrane here have four residues at the very bottom. And E protein also has four residues at the very bottom that should be in very close proximity to capsid protein. And we're looking at the sequence alignment of all of the different flavivirus species, or the most well-studied flavivirus species. We can see that there's quite a high degree of homology within what we call the transmembrane helical turn residue domain, or the TMHTRs. We have transmembrane helix 1 in PRM, so this side here, and transmembrane helix 2, this side here. And those four residues are quite highly conserved. We have a lot of leucines, isoleucines, and valines that all share much of the same chemistry. And then we have glycine here that is conserved across all the different flaviviruses. That seems to be quite important. And then we have a few distinguishing residues between different species at the downstream positions. However, these first two, L146 and G147, seem to be quite interesting because they are conserved across most of the flaviviruses. So my job was to figure out, can we actually understand the assembly mechanisms? And my hypothesis was that the assembly of infectious flavivirus particles is dependent on interactions between the nucleic capsid core and the transmembrane helices of the glycoproteins. And to understand that, I came up with two specific aims. The first being to identify the role of the PRN and E transmembrane helical turn residues, or the TMHTRs, in virus assembly. And in part one of today's talk, we'll be discussing a little bit of the reported virus particle data that I've been able to put forward. And then in part two, if we have time, which I don't think we will, we can talk about some of the fully infectious results. And my second aim was to actually take these residues, take these proteins, PRME, and reconstitute them into lipid nanoparticles to actually look at the results of these hypothetical interactions in vitro. And of course, in today's talk, we'll just focus on part one, but happy to answer questions about the rest of these as well. So to kind of go through briefly some methodology, we have we mutated and the PRME proteins and the RSV and the RVP system. I'll get into that in a moment. What we wanted to do was we did all the mutations, we wanted to understand infectivity phenotype, and that would produce four different phenotypes. If either mutant would be the same as wild type, no change in titer. It could have a reduction in titer, meaning that we're maybe disrupting some kind of mechanism. It could actually replicate the RNA, but it would have zero infectivity. And so that would suggest that we might be disrupting assembly, no virus is being out. And then we also have a lethal mutation. So this would mean that it's not only lethal to make titer, but it's lethal in the fact that no replication is happening. We characterize these mutations using a slew of different methods. Altogether, we made 86 mutants in the RVP system. We had 4 mutants in the infectious Zika virus system using the RVP system as our foundation. And we made a total of 70 mutations within Igntu to confirm the results shown in Zika. Now, again, like I alluded to, we use the Zika RVP system. This is a reporter virus particle system developed by Dr. Ted Pearson, who's the director for the Vaccine Research Center at the National Institutes of Allergy and Infectious Disease here in the United States. What this does is it utilizes the GFP reporter gene directly upstream of all of the West Nile virus non-structural proteins. And we combine this with a second plasmid that contains the structural proteins of Zika virus, so the virus that we are interested in using. And these produce a pseudo-infectious particle. Now, of course, these contain all the structural proteins, Zika PRF, the capsid protein from the structural plasmid, but they also contain the West Nile virus replicon. And what this allows us to do is utilize a single round of replication to identify infectivity, and these viruses are not able to proliferate afterwards. And so we can utilize fluorescent cell counting And this takes our infectivity screen from 9 days to 5 days because we're not waiting for plaques to form, and this helps speed up the process tremendously when dealing with nearly 100 mutations. And so again, to kind of revisit what it is we're trying to understand here, we have the inverted tripod structure, that spike that we looked at earlier, of the PRM, shown here in blue, and E protein heterodimer, shown here in red, in that close proximity to capsid protein. Are these residues right down here at the bottom of these proteins interacting with capsid, or are they helping to develop assembly somehow? And again, those are the residues I'm very interested in. So we'll go ahead and jump into some data here. What we see is first I want to look at the E protein mutations because I figured some of them might have a detrimental effect because how large of a protein E protein is. However, E protein was quite resilient to all kinds of different mutations with only a few mutations resulting in a log reduction in titer and one large mutation for ADW, so a huge tryptophan put right here in the middle of this transmembrane helix, probably broke the helix, which resulted in loss of activity. However, overall, E protein looked very resilient, and this was surprising to me at the time. But PRM showed a much different story. Now, PRM actually showed some quite interesting results here at position 146 and 147. What we can see here is that at 146, we have these two negative residues that almost cut titer in half. I'm sorry, not half, but losing about 4 logs of infectivity in some cases. And then others like this valine at G147, as well as the G147E, another negative residue, and G147B also showed about a 3 log and 2 log reduction as well. Again, we see this phenomenon with large residues being stuck here in the middle, also resulting in loss of titer completely. But what was interesting were these negative residues here. That we had a loss of 4 logs and a loss of 3. And what we see is that L146 and G147, as well as our insert and tag mutants that contain a huge flag tag linkers out of lots of negative residues, actually wiped out titer completely. And so altogether, we're able to kind of put in a list together. We found that 15 mutants, both substitution and inserts, reduced viral infectivity by about 2 to 4 logs. Now, that suggested a few possible outcomes. The first could be that we are actually disrupting virus maturation. And so, unlike the hypothesized results that we could be interrupting assembly, instead we could have been interrupting virus maturation. And so, this is when the virus goes from a spiky immature particle to a mature particle. If it remains in its immature state, it would also reduce infectivity because that state of the virus is not as infectious as its mature form. So that could be one potential outcome. We could also be disrupting attachment and entry. Again, if we remain in that immature phase or if we lock the particle in a specific conformation that does not allow for attachment, we would see a reduction in titer. We could also be having uncoding problems. Maybe the virus is forming perfectly fine, maybe it is attaching and entering in a new cell completely fine, but it is not able to uncode itself. Maybe it's being locked in a conformation again that's inconducive to uncoding. So we would see a reduction in titer. Or, as hypothesized, maybe we are disrupting this cell. And so we needed to do a little bit more homework and find out where within the virus life cycle we are disrupting. And so to do that, we first looked for the amount of E protein that was being released by these mutants. We can see that L146E and D both have about a doubling effect of the the amount of E protein being released into the viral supernatant compared to wild type. And even G147E is still maintaining, but they're 3 or 4 logs reduced in titer. And so all of these E protein being released might be empty particles. And in fact, when we look at supernatant E protein versus RBP titer, we can see that infection is nearly down 100-fold. And so we have about a 2 log reduction in infection here, but we have a 2-fold increase in E protein being released. And so, we have E protein is released, but we know for a fact with inflated viruses that E protein is not just excreted out of the cell. It is a transmembrane anchored protein. And so it is not released unless it is truncated, and truncation only happens in man-made or in human synthesized products. And so naturally, E protein should only be released if it's within a particle structure. And we can also show that E protein is not E protein trafficking from the endoplasmic reticulum to the Golgi to outside of the cell is not being impaired, or else we wouldn't see any. So this suggests that maybe we are producing these things called subviral particles or SVPs. Subviral particles are particles that contain E protein and PRM protein or N protein, but no capsid and no RNA. So pretty much they're an empty shell. They are a shell that is budded off out of the cell that contains no RNA. And it's a very, very low probability of that happening naturally. And so maybe we are, in fact, disrupting assembly of these viruses and releasing empty particles. But we needed to show that we're actually not having the RNA, not just lots of E protein in the supernatant. And in fact, that's what we're able to show. We are able to show that RNA is actually decreased by 100-fold within these purified virus particles, so these mutant particles, excuse me. And when we look at a phenomenon called specific infectivity refers to the number of particles needed to produce a plaque. And so what we can show is that wild type here requires about 10 particles for every plaque to form, whereas these mutants require nearly 1,000 particles. And so we have a decrease in specific infectivity, meaning that there's many, many thousands or hundreds of virus particles that are being released that aren't causing infection due to the fact that they are lacking RNA. And so we are very confident that we can actually say that SVPs are being produced here. But we needed to look even further. And so what we did next was to look at if these mutations could recover titer over time. And we actually see that they don't recover titer over time. And this supports our assembly defect hypothesis, meaning that these viruses kind of plateau off and they're not able to form to recover, so meaning that they're not able to outpace their mutation defect, whatever is causing that defect, whereas wild type was able to peak at a much higher titer. And so this assembly defect seemed to be spot on. We wanted to make sure we could actually maybe try and get into the cell and see what's going on. What is changing? We know that these mutations, these single point mutations of a positive residue to a negative residue results in a loss of infectivity and a decrease in RNA, but why? And so first thing we wanted to do was make sure that these proteins are all getting to the right places. And so what we did was we purified some mutants of this PRM L146E mutants. We purified using about, I think it was close to 40 T175 flasks of transfected cells that were taken through ultracentrifugation steps. Very long, drawn out process, but we made it work. And we were able to actually show differences between the wild type version, shown here in lane 3, and the mutant version, shown here in lane 4. The first one we're looking for is capsid protein. And we can see clear that these two samples both show E protein, but capsid is lost in the mutant version. And what we can see is that it's highly significant, it's downgraded significantly. And now what this suggests was that capsid is in the proper location. And so it should be within the cytosol, and we can see that it's not in the virus particles themselves. And let me back up, I'm so sorry. These are not actual virus particles, these are Golgi samples. These are purified Golgi samples from transfected cells. And we want to make sure that if we are in fact getting proper trafficking here from the virus life cycle, we have assembly down here. We're actually going up, and if the virus assembles properly, it's transferred to the Golgi. That Golgi is then the virus is able to mature, and then it is released out of the cell. And so my apologies. We want to look at whether or not we're getting capsid within the mutant Golgi. And if we are not getting in the Golgi, that would suggest that we are stopping assembly from occurring at the endoplasmic reticulum. And that's exactly what we see here. And here are a bunch of, what's it called, standards, a bunch of positive controls showing that the region that we are looking at, getting some feedback, but I'll keep on going here. We can see that capsid is in fact missing from these Golgi samples. And all of the positive controls showing that the different compartments are in fact intact, and we're not seeing any bleed over from the cytosol. Now, this is encouraging because capsid is reduced in the PRM L146E Golgi samples. And that suggests that assembly is being severely impacted by single point mutations in the PRM transmembrane term region. And this supports our hypothesis that SVPs are being produced. You see here, get some feedback. And we, could you mute yourself? I'm so sorry. Thank you so much. I appreciate it. All right, let me get back in here. Go ahead. Keep on going. All right. However, okay, so if we are actually seeing, so the other major issue that we're running into is if we are disrupting this heterodimerization between PRM and E protein, we would also be stopping the trafficking of the viruses between the ER and Golgi. And so we might not actually be making SCPs, instead we are disrupting the heterodimerization process. And so we had to address that issue before validating the SVP production hypothesis. And so to do that, we of course looked at these purified proteins, these purified RBP samples, and we can see again that E protein is highly visible, but capsid is lacking. And these are indeed the purified RBP samples, not the Golgi like we saw in the last slide. And what we can see is that, of course, using optic density, there's a high reduction in capsid band intensity. But next, what we did was we looked at label-free quantitation using mass spectrometry to see whether or not we are disrupting heterodimerization between M protein and E protein. And what we can see is that the ratio of M to E, both within the wild type and the mutant, were pretty much 1-to-1. And there's no significant change between the two. However, when we look at the PR peptide as well, are we getting a difference in maturation? Because PR peptide is cleaved after virus maturation and it is released after the virus is released from the cell. If we see a difference there, maybe there could be a maturation defect. However, we don't see any difference there whatsoever. And the only difference that we're able to find is that there's a huge reduction in capsid protein. So this suggests ultimately that we are in fact making SVPs. But why are we making SVPs? A previous study published only this past year, 2023, actually showed that the PRM transmembrane helices contain a cholesterol binding motif that is actually right downstream and upstream from the TMHTRs. So these highly conserved residues that we talked about before, there is a cholesterol binding site between both of them. So what this suggests is that maybe our mutation here, PRM L146E, is disrupting some cholesterol integration into these binding pockets that could contribute to the assembly defects. And so what we did was look at the different cholesterol contents of the viral membranes of these purified RBP particles. And so what we can see is that, yes, we actually do have a tremendous decrease in the amount of cholesterol between the wild type and the mutant version. But we wanted to make sure that that wasn't an artifact from the cells themselves. And so using the whole cell lysate, we can see that there's no major difference between the different groups, mock-infected, wild type, or the mutant version, suggesting that cholesterol itself is not being disrupted within these cells. It is only being disrupted in its integration into the virus membrane. Now we can also break this down even further and actually fractionate these cells and look at the membrane back fraction themselves from the plasma membrane. And we can see that there's a decrease in between mock cells compared to wild type, but the mutant version is also decreasing compared to mock cells, meaning that cholesterol is indeed being trafficked from the plasma membrane to the sites of replication and budding, where it is then incorporated into the particles. However, the same trafficked cholesterol that is being incorporated in particles in the wild type version is not making it into the version of PRM L146E. So something else is happening there. So next, looking at the endoplasmic reticulum fraction, this is where the viruses actually bud from the cells. And we can see, interestingly enough, that there is slightly more here. There's not a significant difference, but the power of significance between the mock version versus wild type is slightly significant, whereas the mock to the PRM L146E is a bit more significant, suggesting that that leftover cholesterol is still stuck within the endoplasmic reticulum, so it's not making it into the particles. Our mutations are disrupting cholesterol's integration into the virus membrane. So this seems like the PRN L146E generated SVPs make the SVPs by reducing cholesterol. But could there be more to this equation? We wanted to look beyond cholesterol and see if there's other lipid groups, because there's many lipid groups that are highly important within the virus membrane, if those were also altered. So doing a single particle analysis of the lipidomics of these different proteins, viruses, excuse me, we're actually able to show that about 201 lipids were found to be significantly different between the wild type and the PRM L146E RNPs. And going in a little bit further, we're actually able to show that of these 201, 72 lipids were found to be twofold different than wild type compared to the mutant version. And kind of funneling down these 72 different lipid hits even further, we're able to show that 28 final lipids were found to be significantly different between wild type and the PRM L146E mutant. And many of those were actually ceramides, phosphatidylcholines, and then phosphatidylglycerols, so highly negative lipids as well. Now, those highly negative lipids shown here, we had ceramides, which contain a negative grouping in some of them, and then phosphatidylglycerol were also significantly reduced within the viral membranes. So not only are we losing cholesterol, but we're losing negatively charged lipids as well. All of the other lipid groups seem to be quite similar, so there's no major differences there. However, cholesterol, ceramides, and phosphatidylglycerol were all depleted within the mutant version. And so it seems that PRML146E seems to be discouraging the integration of these specific lipids and steroid groups into the RBP membrane. So this brings us to our ultimate conclusion here. In my original hypothesis, I assumed that the interactions between the nucleocapsid core and the transmembrane helices of the envelope glycoproteins would be a one-to-one protein-protein interaction. However, we've been able to update this hypothesis and change it to better understand the dynamics at play, and that the assembly of infectious slave virus particles is actually dependent on intermediate lipid interactions between the PRM transmembrane helical charge residues and the nucleocapsid core. And so with that, I'd like to leave you with this schematic here that we're hoping to publish this year of what's actually happening during flavivirus assembly. So starting here in the top left corner of the screen, you can see that in a normal wild-type Zika RBP situation, the wild-type PRM transmembrane helices actually encourage the formation of negative membrane patches. So these small membrane patches of negative lipids can actually act as a targeting beacon for nucleocapsid core formation. So as we said before, capsid protein is very positively charged. And so if these PRM transmembrane helices are in fact encouraging more negative lipids to surround them, this would encourage a highly positively charged nature of capsid protein to be attracted to those regions. And so it's not as if the proteins themselves are interacting with capsid, but through those lipid intermediates, they're forming the proper environment needed to attract capsid protein to them. Now, this high degree of positive charge within that inner membrane of the virus, of the budding virus, recruits the viral RNA. The reason we believe this to be the case is because of our SVB production in our mutant version. So up here in the top right of the screen now, we have our mutant PRM transcript helices actually do not promote the negative membrane patch formation. And because of the net reduction of negative lipids. We believe this is caused by the mutation of that positive, the leucine here, into that glutamic acid. And we can see that that negative residue there is discouraging the integration of those negative lipids around the PRM transmembrane helix. And without those negative lipids there, we actually lose the charge needed to recruit those capsid proteins, which does not end up recruiting the RNA, which buds off defected SVP particles. And so this kind of paints a much clearer picture that maybe because of the integration of these negative lipids, we are getting the formation of proper virions. And so if we can find ways of either reducing the negative lipids being made within cells, we might be able to find a whole new novel pathway of treating these viruses that has yet been explored. So with that, I'd like to just say thank you to all of my different collaborators and my old lab members. I'm glad to be within a company now at Radivir to be making drugs against dengue virus. We have some great promising data I can hopefully share with you all in the future. But for now, I can leave you with that and would be happy to take any questions. Thank you so much.
Thank you, sir, for such an informative scientific session. Really, the slides were informative. As soon as possible, we will share you the token of appreciation to you. Once again, lot of thank you on behalf of entire conference team and Department of Pharmacy. I like to request Dr. Devnarayan sir for the vote of thanks.
Okay, everyone. Is there any question? Any question? No. Dr. Nicholas, there is no questions. Good morning, Dr. Nicholas. On behalf of Department of Pharmacy, Indira Gandhi National Tribal University, Amarkantak, India, I extend my heartfelt thanks to Dr. Governor Nicholas for his insightful talk today. Dr. Nicholas, your expertise and passion for your work at David Inc. and Purdue University have truly inspired us all. Your presentation shed light on exploration of the structural and biochemical assembly mechanism of flaviviruses, and it has certainly enriched our understanding of the same. We appreciate the time and effort you took to share your knowledge with us. Thank you once again, Dr. Nicholas, for your invaluable contributions. We look forward to future collaborations and learning opportunities. Thank you. Thank you very much. Thank you very much.
As proceeding forward to our next session, I feel honored to invite respected Dr. Akhilesh Tiwari sir to welcome Dr. Akhilesh Dave.
Very good morning to all. In this wonderful event, today's international conference, I would like to introduce Kapilesh Dave sir. Kapilesh sir has started his career as a teacher, and after some time, he has started his own business, is a extraction plant for some herbal drugs. Now he's having the biggest plant in Mandsaur region. You know that Mandsaur is a big business area, or one of the important business area for herbal drugs in the Asia, not only India. It is number one in Asia. They supply drugs, they extract many drugs, and they export also. So we are really feeling very grateful to introduce Kamlesh sir. Sir,
Yes, sir. Thank you.
yes, thank you, sir.
Hello, everyone. Very good morning to all of you, respected dignitaries, all the fellow gentlemen, and my dear friends. So today I'm going to give you a brief idea of the present status, new horizons, new opportunities, whatever is going on in the field of herbal extractions, and how can anyone join this excellent field, because we all are pharmaceutical, right? So there is a new segment where we can explore many more opportunities. Right. So this is about the new horizons of extraction manufacturing. I'm giving you a brief overview in the frame of my own journey. So going to the next slide. Just have an idea about what is how to establish an industry. Okay, because we are speaking about establishing in reason where you most of you belongs the movement reason, that is how we can. So as a student of pharmacy, everyone you must understand what is the core concept of establishing an industry. So establishing a herbal extraction plant or manufacturing unit comes under the MSME. MSME is nothing but a government body who care of the small, medium, and mega micro industries. Okay, so it is an playing an essential role in all the overall manufacturing sector of India. Takes the in comparison to other countries is 30%. So major constraint in the growth and to finance, access to technology and skilled manpower, access to raw material, access to market and infrastructural efficiencies. Going with an idea of establishing a food related industry over in India. Next please. So the export of herbs and value extracts of medicinal herbs are greatly increasing over the years. In comparison to India, we exported around US dollar 330 million worth of herb during 2017-18. And in the next slide, I will give you the idea of the last 4 years statistics. So it is increasing every year. Also, the export of value-added extract of medicinal herbs during that year was also 456.12 million, they're recording the growth rate of 12.23%. So every year, if you study the 5 year statistics, we are gradually increasing our export from 12 to 20%. Every year it is increasing day by day. Next, please. These are the statistics of 4 year data. I'm just giving you an idea because these figures will motivate you to understand about the capacity and capability of herbal extraction market. What world is needed now after the COVID? Everyone is running towards the herbs and nutraceuticals. The market is changing the name from herbal extracts to now it is called nutraceutical, health ingredient, food ingredient, and all these classic words are getting introduced and nurtured by the Indian system of medicine. This is our Ayurvedic system, this is our herbal extracts, this is our pharmacognosy department,
So somewhere in many ways, we all are very much attached with the domain. So if you see the figures from 2015-16 to 2019-20, it is gradually increasing from 274 to $465 million. And specifically, Ayush related products also from 364 to 560. So this is a good growth rate which motivate many of the industry person, many of the ideation person, many of the students of the field of herbal extraction are also small to share other departments. So in the next slide, I will give some more ideas. So from Germany, there are 700 plant-based medicine. Some kind of act. Sorry. In India, it is still it is competitive in the terms of the for the allopathy system in nutrition which are having this kind of herbal nutraceutical ingredients with the with support to support the treatment, oral treatment. And the US increasing public dissatisfaction turning to natural or organic remedies has led to an increase in the herbal medicinal use. Presently, 70% of herbal export from India is going to US only. If you see the data, out of the 100%, if we are exporting 100 tons, that 70 tons is going to US. And our own consumption is only 3% in comparison to US market. So despite a steady performance of this sector over the years, India's share in the world's herbal export is only 1.6%. So who is playing the major role? We know that herbal extract or medicinal plants are a core property of India, but no, China has took over us, and the major stakeholder is China and USA now in the field of extraction. So global market of MF is largely dominated by China, Japan, France, Germany, Italy, and all these countries. So you see the Ayush data. Ayush includes Unani, Siddha, homeopathic, and Ayurvedic. Out of these four, our major shareholder is the Ayurvedic medicine. And these are just the number of manufacturing units. Okay, just giving you an idea of how we are growing day by day, or year by year, or decade by decade. Okay, moving to the next. So the export quantity of export medicinal plant, including plant extract, was 57.880 metric ton. Okay, and the Xylem husk, this is the Isabgol is the major stakeholder right now. Total value of plant raw drug exported was around 354 and corresponding to quantity of 56,500 metric ton. The annual export of India's herbal sector added to the 807 crores at per DGCIS data. Why I am giving you these figures? Because these figures motivated me in the early stage to get into this field. Okay, so these are some figures. Also, we are importing now because our own market also needed some of the medicinal value-added plant-based components in the field. So we are also importing from China. We are also importing. We are importing from Indonesia, Malaysia, Japan. Okay, so we are also importing. So total 40 commodities we are importing right now. The consolidated figure is 37. export. And out of that, gum oil is a major constituent which we are doing. And the next slide, you can get the which is a sample of it, ashwagandha extract, which is majorly we do from the center of the India, from Manson image region, which is known worldwide for the best quality of ashwagandha in the world. So these many kinds of ashwagandha extracts with us. So that is our expertise. We think of about the what kind of herbs available around to you in the larger quantity. Because once we think about the industry, their volumes are pretty high. Okay, so we cannot start our things with a small quantum. So that is not so an industry. If you are planning to do that, there are information given about the MSME site. We just go through that. There are three: micro, small, and medium. Micro having the investment size less than 1 CR. Small is having investment less than 50 CR, and more than 50 CR that comes in the medium. So 50 to 250 crore is coming in the medium, and after that the industry is considered as a large industry. They are not considered as a MSME part. What required to start an MSME unit? There are 4 P's are required. First, you have to decide the product, then process, then the place, the location, and the partner. Okay. So these are the kind of raw material in your area, and also your expertise to do extract, a powder, or isolate new nutraceutical molecule. The R&D part is a huge requirement in that sector, because in previous time, whatever written in our old Ayurvedic data, that was considered, but now the pharmaceutical fraternity involved in that, the regulatory involvement is increasing day by day. USP, BP, IP, and all the pharmacopoeias getting introduced there in standards. Okay, so here, let's see. Considering the pharmaceutical field is related to completely synthetic molecules, whereas in herbal extraction or in nutraceutical industry, there are products which are derived from or extracted from or isolated from the natural origin plant-based medicine. Okay, so product is very much important. Now the steps, first you have to identify the opportunity, what I said to you, you have to study the survey, what is the herbs available around you, then just select two product of your choice in which you are having your expertise, and select the process. There are a lot of processes available for the extraction. For that, you have to be very much specific while your courses, because the extraction processes available in your books can be converted in the frame of large manufacturing setup. Okay, so these are the three parameters which are related to your skill. The fourth parameter related to your financial capacity. You have to arrange funds. I will give you some idea of the figures of finance which you need to require before starting industry. Okay, so then you have to go and identify the vendors who can supply you the plant and machine. They are designed as per your specification, or you can hire some consultants also. They can arrange it for you. Then building construction, you is an area where you can spend more or you can save more. Okay. Then getting the utility connections and all the facilities which require water is required, electricity is required. Then new machine material, and this is an also requirement. Whatever technology is getting introduced every day, whatever kind of herbs are getting introduced every day, what kind of molecules are getting introduced every day by the
then the licensing. In India, licensing is a tough but transparent process now. Okay, so you have to be specific with the requirement. I'm just giving you the brief idea because in late you must be if you are an informed student, you must be possess legal extraction if you are from the field of pharmacology. But usually not involved in the extraction. One analytical part is it starts from the collection of raw material. Then you need to do material identification with the help of PLC, which you are already doing during your BCom studies. Then you need to get it approved from the QC lab on the basis of all the physical chemical properties, so that at the time of your exploring you may get benefited. Then sizing of raw material, sizing and the particle size reduction, what role is there? That is a separate field because this small segment is pretty much important in the field of extraction. Okay, then the calculation of weight loss, production area to supply, then checking of material for loading, equipment cleaning, loading of material for extraction. So there are a lot of extraction approaches available. It varies with the with product to product and what you require. So sometimes it is hydroalcoholic, sometimes with the help of a hexane, sometimes it is from ethyl acetate, sometimes it is from chloroform. So many of the chemical from the polarity basis we select to get the correct extract what we needed. Okay. So if you are interested, just go to your pharmacology and college teachers. They will give you a brief idea what is the polarity of liquid, which help you to decide the solvent system for your extraction media. Okay. Then multiple washing because this is an important part because right now European Union and US FDA both putting every day small checkpoints. Okay, so for toxin, for pesticide, for ethylene oxide traces, from pathogens, for microorganism, for heavy metals. So during the process, because it is not possible to get a raw material which is free from all this pesticide, heavy metal. Because in India, practicing cultivation of farming is still a challenging task. We are not getting herbs which are clean. Okay, so you will get the herbs with all this pesticide, all this ETO, all the traces of microorganism and heavy metals. You have to remove this all through the process. So here your knowledge will play an important role. How will you perform that? That is an R&D part that you can do while you are BCom M.Com studies. If you do that, that will help in future. Okay. Moving to the next slide. After multiple washing, clear pesticide and toxin, the extraction starts with the help of specific period of time is an important three factor: the time, the temperature, and the pressure. Time, temperature, and pressure, along with the suitable solvent system, will give you the exact amount of extraction which will have your therapeutic ingredient in that. Okay, so extraction then goes to the distillation. Distillation after that distillation is also an important part because there are so many types for dryers available. In your laboratory, you also see the drum dryer particularly is having that kind of facility. So there are the basic version of dryers. And if you're going to an analytic tool, so that is a class assembly you will find in your lab. Alright, assembly. So spray dryer, drum dryer is all research in data analytical science. Okay. And then the collected raw material will go for the grinding. Again, the particle size what? Because your client or industry needed different kind of size material, particle size material that is on the 40 mesh, 20 mesh, 60 mesh, 80 mesh, 100 mesh. And then the require also for that you need to go for the granulation. Granulation is an important part. You all must be doing
if you are from the semester 7th or 8th or 6th, and during the tablet preparation, you all must have taken consideration or taken planning for the granulation process. That granulation also playing an important role over here. The herbal extract granules is the current demand. Okay, so after the collecting this fine powder or granule, you go for the quality analysis. Once you get okay, it's the design of a full extraction plant. Okay, so this is a continuous batch process. If you understand that, you just feed the raw material, then it continues for more than 24 to 48 hours. Okay, this is a brief design. The area, whatever requirements, design specific, anyone consult to me. Okay, I will give you an idea and will give support. If you are interested to establish your own industry, but this is not a small project right now. Okay, because US FDA getting involved in that, Indian government is also getting involved in that. So in the next slide, I will give some list of plants and history which they. And the majorly it is having the SS unit for the extraction, distillation, and drying. And all the system are well connected with the help of supply chain straight and get you the correct completion of. These are the power supply needed for each and every equipment. Just we are having less time. I am not elaborating it much more, but you can go through it. The slide will available with your institute. Okay, the schedule which we require the next slide please. Once you start establishing the industry, it will take 9 months if you are pre-prepared. Okay, whatever points I have told you earlier, you have to do R&D, you have to do research, you have to do analysis of the facts and figures, and the basis of that you prepare your master plan. And once you start ready with your master plan, it will take around 6 to 9 months for the implementation and start of the production. Next page. Then I told you. Okay, so basically industry is run in the three shift. That is 88 hours. Um, for each shift. Okay, so industry run for 30 days in a year. Power requirement for the local extraction is 90 kilowatt, 200 kilowatt. The land area required for a standard size of extraction unit is 50,000 square feet, and the cost of installation and erection is around 20% of your machinery and equipment. Okay, next please. The project cost. Overall project cost is comes around 10 to 15 CR. Okay, the reserve fund is 5 CR, 5 crore. There are so many different schemes of subsidy available in CR. You will get about 50% subsidy from the PMEGP scheme. If your product size, project size is more than 10 crore, then you are not eligible for any kind of subsidy. So just try to keep your project below 10 crore. So you will get 40% subsidy from the government of India. That will come in 4 equal parts every year. If you are registering your company as a private limited or LLP or partnership firm, you can get in for 3 consecutive years. You will not get you will not need to pay any income tax. So tax assumption is provided by the government of India if you are private limited or a LLP company. So National Medicinal Plant Board also support many schemes of our cultivation and collection of herbs. If you are in your area specifically, you must, if you are interested to start that, you must contact National Medicinal Plant Board. Okay, so you just need to prepare a budget. You need to estimate the revenue, the fixed cost, variable cost, one-time expense, cash flow, and profit. These are the core part of the financial planning. Before starting an industry, before starting any manufacturing business, you have to assess your financial figures in these 6 parameters. Then only bank will also support you. So the funding also provided. I'm giving brief data of overall project cost calculation in the next slide. And this having the fixed asset expenses which comes under the plant and machinery and building, and the running capital which require for the working capital, which is required for the day-to-day expenses. So every month you need around 50 to 60 lakhs, including raw material and the running expenses, and the overall project cost is 10 CR, which comes for the fixed asset, that is land, building and machinery and equipment. Okay, so 50 lakhs per month day-to-day expenses and the 10 crore. So that's why I told him earlier, because for this kind of project you need 15 CR funds in your hand. In your hand means bank will also support you if you are having collateral properties, if you are having land and constructed building like your home or shops, whatever is there, bank will give you payment against that. Then few certification next please. Few certification and approvals also required. These are first you have to go for the MSME registration, then your firm registration, GST, FSSI, Ayush license, NOC from the Policy Board, license for the solvent use. Then you have to go for Ayush Excel, SEP Excel, GMP, ISO, Halal and kosher certification. Ayush Excel and SEP Excel are the two central government bodies which help you to export your drugs in the foreign countries. So Ayush Excel will give you the support for the participation in all kind of conference, seminar, and trade fairs over there. They are also funded for that. And SEP Excel will approve your product. They issue health certificate for each and every consignment which is going to be exported from India to, especially to European market. Okay, so finance which we needed, your own fund needed to be 30%. If your project costed 15 CR, so you must have 4.5 CR with your hand. That might be from your friends, family. They can invest with you. Then 70% bank loan you can get if you are having property of 150%. Okay, so if you are going to take bank loan for 5 CR, you must have a property value of 7.5 CR. The threats, just going to finish in 1 minute. The total world, next please. The threat means what will give you pressure to do this kind of activity. So you know it is increasing every day, but India's share is global export of herbal products is low due to the following reason. Main reason what are the behind that? That is inadequate agricultural practices. We are such a big citizens are involved in the farming, but they are not practicing agriculture or they are not cultivating herbs in proper way. So inadequate quality control procedure available in the market, lack of large scale organic cultivation, lack of processing and R&D, lack of standardization in product process and services, and lack of regulatory framework in trade of medicinal plants. Okay, but what is the hope after this? The hope we can say that we are having a vast knowledge of Ayurvedic medicine. If we value our knowledge and like you pharmacy people who are studying well, okay, they can utilize the curricular knowledge, just converting it to the industrial knowledge, that will help you a lot. If you're not able to be a part of establishing an industry, you must be a part of the team. Okay, you may be a pharmacist. As a pharmacist, you can join over there. As a research scientist, you can join over there. There, as an analytical chemist, you can join over there. There are so many openings available. Still, even I personally searching many of the person who can having brain of analytical thought process. If you are having that kind of concept, if you are having that kind of ability with you, that will help you to be involved in this great field. If your area is having good quality forest reserve and organic agricultural practices, everyone around you in the village, in the nearby places, just motivate them to do organic agricultural practices, because for 2 3 years they may be suffer for payment return, but after that they will get good benefited from that. So what leader in the field of forget we all have to believe in our own the history of the knowledge of herbal medicine, the Ayurveda, and the practicing medicine in India. Okay. So this is all about a general idea. What I can give it to you from my learning since last 5 to 6 years. I have spent many of the time in the academics, and after that, since last 5 years, I'm in the field of herbal extraction, and we are exporting now to Europe, USA, and Australia. So I learned these things. I just wanted to share with you, all the students and faculty members. Just think over it. This is the classical field where you can explore your ideas, where you can explore your imagination, the abilities, and the innovation mindset. Okay, so thanks you all. Thank you, Adrian. Thank you, faculty members, for giving me this opportunity to share my views and ideas with you. Thank you all. Thank you, Ken, for such an informative scientific session. Any questions? Yes.
A very warm good morning, sir. Myself Aditya. Question is, can you briefly tell us, is there any new advancement in the herbal drug extraction technology? Thank you, sir. Can you repeat, sir? Am I audible, sir?
Yes, sir. Yes, sir. I'm getting your word.
I mean, again, 15% question, sir. Can you briefly tell us, is there any new advancement in the herbal drug extraction technology? Thank you, sir.
Sorry, not getting clearly. What you want to know?
Sir, is there any new technology came?
Technology. New technology. You want to know
Yeah.
about new technology?
In the extraction.
Yeah, actually, presently, here we do solvent extraction. Now the new technology is coming around, that is CO2 based extraction. Okay, so this is the current age where a lot of the new research is going on, but still the world needed is we need to go for the isolation of active pharmaceutical active herbal ingredients from the herbal extracts. So more research in that field is weighted over this time.
Thank you, sir.
Yeah. Any further questions? Yes.
Good morning, sir.
Morning, morning.
Sir, my question is that, sir, can you tell us about your organization working on novel herb formulation and your future plan about that?
Presently, we are working in B2B model where we do extract and supply to world's great manufacturers. We are presently not doing B2C. What is different between B2B and B2C? We are doing business with businessmen. We are not dealing directly with the customer. But next year, we are going to introduce our own formulations. Our research team is working since last 4 years. So we are almost ready with the 10 to 12 formulations which are having some good quality extract specification. So that are going to introduce in the market in next year. So we are going to launch it worldwide. In more than 40 countries, we are in the process of certification and approvals. So once we are ready with all the certification, testing, and clinical trials, especially clinical trials, and then we will introduce our products to market. So here I would like to mention to that the institute which are having facility to perform preclinical studies, the industry is looking for tie up with that kind of institutions. Okay. So as a student, as a faculty member, or as a department, I suggest everyone to classify or develop their own facility to industry friendly. Okay, if you are having the culture in your department, industry will directly come to you and you will get involved with all the modern practices going around you. Okay. So these are my planning. I am presently doing extraction only. We are having more than 60 kind of herbal extracts. We are having our own dedicated unit for organic product that is having around 14 products in hand. So in 14 organic product and 60 kind of regular herbal extracts, we are presently doing almost 75 kind of herbal extracts right now. And next year, hopefully, we will come to you and market with our research-based pharmaceutical formulations which have herbal nutraceutical ingredient in that. Okay.
Okay, sir. Thank you, sir.
Yeah. Thank you. Any further questions? Okay, no questions, sir. Thank you so much, sir, for such an informative scientific session. As soon as possible, sir, we will share you to the token of appreciation. Once again, lot of thank you on behalf of entire team and Department of Pharmacy, IGNTU. I like to request Dr. Rishi Paliwal. I like to request Dr. Rishi Paliwal, sir, for the vote of thanks, sir.
Very good morning, Dr. Prakash. And it has been yeah, it has been really wonderful lecture, and I understand from the industry point of view, you have shared all of your experience, and it has enriched our the theme of the conference definitely. And a lot of students are here. Some may be entrepreneur in future, they might be planning for some industrial scale up of the products or such kind of things. So you have very beautifully delivered your talk in very short time. We will definitely be in touch with you at other platform whenever it is required. Your services, your expertise, sometimes offline. So thank you being with us on online, and I know it is a busy schedule. So on behalf of entire Department of Pharmacy and organizing team, I acknowledge your contribution and thanks to you. It has been really wonderful. Thank you so much. Thank you.
Thank you so much, sir.
सभी को धन्यवाद करना चाहूंगी आपके समय के लिए और अद्भुत सहयोग के लिए कृपया जोरदार तालियां सभी के लिए तो दोस्तों कुछ ही पलों में हमारे मुख्य अतिथि हमारे बीच होंगे इस कार्यक्रम में होंगे तो मैं आप सभी से अनुरोध करना चाहूंगी कि जब उनका आगमन होगा तब आप सभी अपने अपने स्थान पर खड़े हो जाएं और उनके आगमन पर जोरदार तालियों के साथ उनका स्वागत करेंगे कृपया अपने अपने मोबाइल फोन और इलेक्ट्रॉनिक डिवाइसेस साइलेंट मोड पर डाल दें ताकि कार्यक्रम के दौरान किसी भी प्रकार की बाधा न